Yeah, that's okay. Don't get too and you guys know how to send it to the meeting, right? Oh no.
Congratulations on your abstract getting accepted.
Oh yeah.
That was awesome.
Okay. By the way, Miriam, thanks for the
Yes.
hat. Thank
You're
you for
welcome. I
that.
hope it keeps you warm.
It did today, for sure.
It's so cold.
Are we good?
You good to go?
Yep. Actually work today?
Hopefully, I think one tried to turn. We just checked the stream. It is streaming from the link I sent you, so it should be okay.
Is it PC1 for your name or is it
PC1 is the...
Sounds good.
Apologies, are you sending your file to Yeah, if we can find the floor, yes. All right, folks. All right, we have quite a lot that we need to cover today, so I hope that you did have a really good reading week break. And we are now kind of going to go through things that you need to know. There are a couple of other really important announcements that I'm going to go through as well. So if you have any questions at any point in time, please let me know about that. And we are using Notonic. If you are logged on, you should start to see this poll coming online for you. We're also hoping that there is going to be at least 200 of you in class today. If we do pass the 200 in-person attendance or online and in-person attendance combined, I will give you like one of your final exam questions in advance. And for every class that we have over 200, we will have like one question in advance. So we'll see how we do today. All right, so before break, and I hope that you had a really good one, we were talking about a lot of different things that I'm indicating for you up here on this particular slide. So we had been looking at some of the different ways in which maybe things like diet might be able to affect conditions like depression. And we know that there is also a link between the gut microbiome as well as through the vagus nerve, maybe activation of different immune cells within the brain, and we might be able to, through these short-chain fatty acids, alter some of the function of either microglial cells or potentially even directly the neurons themselves. Today, our focus is going to be slightly different. We're going to be looking at this system where we have looked at the effect of omega-3 fatty acids in the diet, and we're going to take a look to see if there's going to be the importance of these other immune cells, and I'm going to be talking about these immune cells off and on. I'm just going to also point out things to you on this slide as a review, and if you don't remember any of these different things, then maybe you should circle them, come in for office hours, and make sure that you're okay with them. They will probably be on your short answers. So things like LPS, lipopolysaccharide, we used that as a model right before we took a break, and you should know what LPS is, why we were using LPS, and why it might be important. So again, hopefully it's a little bit of a review for you, and if it's not, it's okay. You have lots of time to kind of make sure that you're going to be in an okay situation. So this is also one of the pathways that we talked about right before break, and I wanted to go back and talk to you about it because this will be a really big part of our story a little bit later on today. We had been talking about the ways in which different forms of stress, whether it's chronic, unpredictable stress that we use in models, or psychological stress that you're all affected by, can impact on the brain. And in particular, one structure that is very relevant for you as students when we're looking at models for depression is the hippocampus. The hippocampus is affected quite severely by things like stress. And one of the things that we saw in the last lecture, hopefully as a review for you, of all the things that go down in the brain following depression, and many, many different things go down, things like BDNF levels go down in the brain, VEGF levels go down in the brain, neuropeptide Y is not something you need to focus or worry about, and we know that some of the signaling pathways, like the ERK kinase pathway, also becomes less phosphorylated and less activated, so this phospho-ERK 1-2 pathway, we see a reduction in the signaling. But strangely, in certain instances, we see an increase in phosphatase activity like MKP1. Do you remember MKP1 in this pathway that I'm showing you here? And some of you are asking, like, what turns on this MKP1 pathway? And again, it's not really clear how all of this works, and this is sort of a diagram where maybe some of these pathways might be important. And maybe some of these selective serotonin reuptake inhibitors, these antidepressants, can directly block MKP1. It's a theory, it's a hypothesis. No one has actually directly shown that yet. And this is important because it is blocking the phosphorylation, and this is why we see this inhibition. If we have MKP1, it's going to inhibit this ERK1 phosphorylation activity. This will block the dimerization and the phosphorylation of CRAB. It won't be able to activate through the cyclic AMP response element. It won't therefore be able to turn on the production of BDNF, VEGF, and others. And previously, I didn't go through this other pathway where stress might somehow, through mechanisms that we don't fully understand, through the MAP kinase pathway, alter the activation of the transcription factor ATF2 to increase the production of MKP1. Those question marks tell us we don't know all of the pathways. Classically, we know that ATF2 does lead to the production and the transcription of factors like MKP1. So this is an important slide. You should make sure that the different pathways that I'm showing to you here on this slide are ones that you are going to be comfortable relaying back on the final exam. You should expect that there will be both short answer questions, like one-line responses. So if something is going up, what do you predict will happen to the rest of this pathway, as well as a potentially long answer question that's based in part on this one slide here. So it is an important slide. I want you to think about it in your recognized study groups or to discuss it with me during office hours, but hopefully some of it, not all of it, but some of it is a review for you. So here are some other things to keep in mind, and this is just for your reference, and the slide here on the left isn't something you should worry too much about, but the other parts are a little bit more important. So one of the biggest things that I've tried to get across, whether it's through diet, whether it's through the different types of drugs that you can take to treat depression, it seems to work in an anti-inflammatory pathway. All of the different things, whether it's like omega-3s, anti-inflammatory, exercise, anti-inflammatory. Some of the SSRIs may be anti-inflammatory as well. And so these anti-inflammatory agents directly might be able to safely alter some of these depressive symptoms as well. And it's an interesting hypothesis that maybe by taking anti-inflammatory agents, you can directly reduce these depressive symptoms. And one of the reasons why we're particularly interested in this, the cells that we see activated and in the brain after major depression models have set in are these microglial cells that we spent some time on in the last lecture. And again, just as a review, as a recap, to make sure that you're spending the time every week to get ready for the final, remember we have like the M1 and M2 microglial types. And these M1 and M2 microglial types are really important. We see an activation of the M1 type in inflammation as well as in major depression. And we think these microglial cells are also really important in depression as models as well. Here's another reason why these microglial cells are important. They also produce a number of pro-inflammatory cytokines. This might be the way in which inflammation occurs in the first place. And there's this really interesting study that came out on October the 22nd in Nature that actually looks at the role of these microglial cells. And it turns out these microglial cells are much more important than we used to think. It's not about neurons. Neurons are important. They make up the way in which we integrate circuits. They're really important for strengthening, learning, and everything else. But these microglial cells that we've ignored for like well over 100 years, they're now coming back into importance. And I wanted to indicate that to you. If you take a look, if these are all the different types of microglial cells that you see in the adult brain, so we begin with precursor cells embryonically, and again, everything here on this side of the slide is important. And as we develop from an embryological stage, and we become an adult, there is about a third, 30% to about 33%, or 1 out of every 3 of these microglial cells are special microglial cells. And it turns out that these microglial cells are important because they come from a lineage that expresses this protein or this gene known as HOXB8. And these HOXB8 microglial cells tend to be incredibly important. We've never really examined them before. And one of the things that this paper is showing is that if we have a knockout, we eliminate microglial cells so that they don't express HOXB8, then one of the things that results, these animals develop spontaneously by not having microglial cells that don't have this protein, they actually develop obsessive compulsive disorder, and they also develop anxiety. Just eliminating these microglial cells. Everything else is intact. Not all microglial cells, by the way, just the ones that contain HOXB8. The other thing that's important to activate and turn on and to mature these different microglial cells, we need another factor which is shown here, which is known as CSF1 receptor, or CSF1R. And this is colony stimulating factor 1 receptor. And these are found on these microglial cells. And if we don't turn them on, we will also develop this OCD and anxiety. So we need both HOXB8 and we need the receptors for their maturation, which is CSF1R, colony stimulating factor 1 receptor, on these microglial cells in order for us to not develop OCD and anxiety. If we knock them out, these mice develop OCD and anxiety. For those of you who have been taking psychology courses, do you know what this term here at the top is? Any idea? In psychology? Abnormal psychology classes? Yes. Hair pulling, right? So you can tell I've done this type of behavior before, lots of hair pulling. And if you look at someone who has OCD and anxiety, they engage in a lot of this hair pulling. They will pull hair out. And this particular type of behavior is also shown in this mice. Knocking out these microglial cells and increasing inflammation in the brain as a result, through other compensatory pathways, leads to OCD behavior and anxiety. And it's amazing that these microglial cells have such an impact on our brains. And I wanted you to be aware of that. So again, really, really cool study. These mice eventually become so obsessive compulsive as well as anxious that they become completely nude. They pull like all their hair out as they get older. So again, pretty remarkable study. I do want you to make sure that you are familiar with the study shown over here on the right-hand side and some of the details that I mentioned. Some of these molecules like HOXB8, you need microglia with HOXB8. If you delete them, they will develop OCD. You need this for maturation. If you don't have this factor and the receptor for the factor, you will also develop OCD as well as anxiety. I won't get into the sex effects as well. More of the female mice were affected than the males, but that's a hormonal thing that was found in this paper as well. So we were talking right before break about these omega-3 fatty acids, and these omega-3 fatty acids have been implicated in maybe being protective, maybe being good for people with anxiety or depression. And yet again, another study has come out to actually show the exact opposite. So these don't work. Taking omega-3 fatty acids actually have no benefit. They're not protective. They won't help you if you have depression or anxiety. So based on this, if you were asked, how would you evaluate the literature in terms of this study shows one thing, another study shows something completely different? What kind of study would you look at to try to convince yourself one way or the other that omega-3 fatty acids have an impact on depression or not? So you would do a meta-analysis, and this would be one of the papers that you would include that actually show that there is no effect. So again, making sure that you are aware that for the final exam, being able to interpret like a meta-analysis study will be important. And again, does diet really have any effect on depression? It's a completely unknown field. There's like data saying yes, it does. There's also a lot of data that says that diets, different types of diets, have like no effect whatsoever on depression. This particular study that came out recently that's summarized in the Atlantic article shown here actually suggests that certain types of healthy diets are protective, and other studies have shown completely opposite effects. There's no effective diet. And does exercise actually help with depression, which is what we're slowly moving toward. Any questions on any of that before I move on? No? Okay. So if we were to go back and revisit some of the things that we were looking at this particular slide and we were rethinking our model. So how do antidepressants actually work? And one of the things that I've showed you on that earlier slide is that maybe antidepressants don't really work in the way that we think by increasing neurotransmitters at the level of the synapse. And maybe one of the things that it might do is to work in ways that we hadn't thought of directly. Maybe they somehow affect the transcription and the level of BDNF. Maybe they alter the levels of MKP1, or maybe they alter other molecules. And I'd sort of gone through very quickly VEGF, or vascular endothelial growth factor, as well as BDNF, the one molecule that we're going to spend a lot of time on. We know, for example, that stress will decrease the levels of VEGF. It will also decrease the levels of BDNF, and antidepressants tend to increase the levels of VEGF, BDNF, other growth factors like insulin growth factor 1 and fibroblast growth factor 2. All of them work through these dimerized receptors. All of these dimerized receptors, so they always bind them together. There are two of them. This will be a common theme. And this eventually leads to these tyrosine residues being phosphorylated after these molecules bind to the respective receptors, and it leads to all those pathways that we talked about earlier. And if we don't have them, if we have a decrease in some of these factors, we will actually see the neurons shrinking, we'll see their dendrites shrinking, we'll see a loss of neurogenesis, and we will end up with things that look a lot like depression. Antidepressants work in the opposite way, so they may actually increase neurogenesis, the number of neurons that we actually see, so we have more of these neurons in the dentate gyrus. We will increase the size as well as the number of dendrites that we see within different parts of the hippocampus. So we know that these factors are really important. And we know that stress and antidepressants and maybe other factors work against each other or tend to work in opposite directions. So any questions on this before we go on? Or anything that I can clear up for you now, or any questions that remain from last lecture? Yes? Right. So when we have these two, so this is one part of the receptor. So this is one receptor subunit. This is another receptor subunit. These dimerize, and the fact that they dimerize, they will only dimerize in the presence of their ligand, the protein itself. And when they dimerize, they autophosphorylate. As soon as they come together, it's like shaking hands. That causes the phosphorylation of these residues here, these tyrosine residues. Any other questions? Yes? Sorry? HOXB8 is one of the homeobox-related transcriptional regulators. So remember from your bio 130 or 230, you learned about homeobox genes. So it's just a homeobox gene, and it's a transcription factor associated with the homeobox genes. Any other questions? Okay. So let me ask you this question. Do you think you need to know this slide or not? Yes? Anyone else agree? You need to know this slide? Sorry? The subgranular zone. Subgranular zone. So here we are looking at the dentate gyrus. So this is the dentate gyrus. This is where we normally have neurogenesis, where we get in the adult, we think, and especially in adult animals, so mice, rats, we see this all the time. In the adult, within the dentate gyrus, we have all of these different layers within the dentate gyrus. Hopefully it's a review that you've learned in your second year neuroscience courses. You will have structures down here that are part of the hilus, the subgranular zone layer, the granule cell layer, and the molecular layer. These are all different layers. We tend to see the cell bodies concentrated within this granule cell layer. And where we see the beginnings of neurogenesis is in this subzone under the granule cell layer, which is known as the subgranular zone. So it's under the granule cell layer. Does that make sense? And where neurogenesis starts is by these interesting looking cells, which are called radio-like glial cells. And these will spontaneously divide throughout your adulthood into these progenitor cells. And these progenitor cells, in turn, will form these neuroblasts. At every stage, they become just a little bit more differentiated. And these neuroblasts are actually interesting because here, they look a little bit more like neurons. And one of the reasons why they're called neuroblasts is, unlike these progenitor cells, we actually start to see the development of these different GABA-A receptors. Now, these GABA-A receptors at the beginning of this developmental period are very different than the GABA receptors we would see in the mature neurons, and I'll get to that in a moment. These neuroblast receptors, as they continue to differentiate, so one way to look at it is we see different cell types from left to right in the granule cell layer. Another way to look at it is each of these stages represents a different time period for this cell. So this might be the first day, this might be like day 3, again, this might be 4 days later, etc. So these are different time points. And you can see that they stay in this lowest layer to begin with, and they will slowly migrate up to the very top of this granule cell layer. And the other thing that you can notice is that at this later stage, where we start to see the projections of these dendritic-like structures, so we start to see things that look a lot like dendrites, this tree-like extension, is where we will start to have the synapse form between these other interneurons that are releasing GABA. And these inhibitory interneurons are releasing GABA onto these immature DGCs or dentate gyrus cells. And these immature cells are receiving GABA, but instead of hyperpolarizing, GABA here actually works to excite these cells. So it's working in a very, very different way. And one of the reasons why it works in a different way is if you look very carefully, and I didn't show you this at the outset, when we look very carefully, we see these different types of transporters that are associated with these different developmental stages. So early on, these neuroblasts and these immature dentate granule cells that you see here, they will have this NKCC1 transporter And these co-transporters, as the name suggests, move potassium, sodium, as well as chloride, and they actually create a chloride gradient, but in the opposite way that you have traditionally learned it. So remember, chloride is usually higher outside the cell than it is inside the cell. And that's important, but here in these immature cells, there's much more chloride inside the cell than there is outside the cell. And that's because of this particular transporter. Eventually, as this progenitor cell continues to develop, it will eventually form this fully mature granule cell in the dentate gyrus. And you'll see that it has switched and is now showing the adult version of this, which is KCC2. It's switched from this immature form, NKCC1. So we see NKCC1 here. It's starting to begin the process of switching over. And then the fully mature version has this KCC2. And as your colleague pointed out earlier, there are going to be a couple of questions, both one-line short answer questions related to this slide, and when you might expect to see KCC2, or when you might expect to see NKCC1, what type of cell it might be associated with, and there will also be like a multiple choice test question based on this diagram as well. Eventually, by the end of 3 weeks, we have this radioglial-like cell forming this progenitor cell, and over the course of 3 weeks, we'll form this fully functional, fully mature version of the dentate granule cell, and it will make connections to the perforant pathway. It will also send projections down and stimulate the CA3 groups of neurons. Do you have any questions on this or anything that I can clarify for you at this point? And again, just keeping in mind, we talked about this pathway because this time frame of about 3 weeks actually fits with how long it takes for an antidepressant to typically work. So you start taking it, you won't feel any benefit until about 2 to 3 weeks later. Yes? Yep. It is a fully mature neuron. It is fully mature, fully integrated into the hippocampal circuit. Yeah, and it's somewhat controversial, so just going back to try to answer your question in a little bit more detail. These antidepressants are thought to do two different things. One, they're thought to enhance neurogenesis. So we get more of these cells being born in the beginning, so we would actually start having more of these IPC cells, more of these progenitors with antidepressants. And we would also see this time frame, we'd have more of these neurons coming online. Again, we're not really sure what happens under stress. Maybe there's a lack of neurotransmitter. There are a lot of different theories, but that is sort of the theory that's out there right now. Any other questions? Yes, Miriam? So from going to the left side of this diagram, from the progenitor cell or IPC, all the way to this green cell, which is the fully matured dentate gyrus granule cell, it would take about 2 to 3 weeks. Any other questions that I can answer for you now? Yes, Isis? So it's just a type of intermediary progenitor cell. So we're just calling them progenitor cells, okay? Any other questions? Yes? Correct. Yep. So again, the immature version of the NKCC1 versus the mature version, the KCC2. And so this is the potassium chloride co-transporting molecule. So that sets the normal sort of high levels of potassium inside, high levels of chloride outside, the thing that we're traditionally used to. Okay, so within the brain, this is sort of an interesting phenomenon as well. What we've been looking at is within the hippocampus, particularly within the dentate gyrus, we see a lot of this neurogenesis going on. Another hotspot for neurogenesis within the adult brain is in the subventricular zone layer, and I mentioned that in passing. And again, we don't know if depression is affecting all of these pathways directly.
We think that there is also some neurogenesis occurring within the hypothalamus. Very, very controversial. Some scientists believe that there is. Some scientists believe that there isn't. These are the only two that everyone agrees occurs in the adult brain. Hippocampus, dentate gyrus, 100% everyone agrees. Subventricular zone, 100% everyone agrees this occurs. Hypothalamus, do we see neurogenesis occurring? It's about 50-50. Some scientists say yes, some scientists say no. It's all artifactual. Does it actually occur in humans? And again, this slide is not one that you need to worry too much about. In humans, different areas have also been identified. Similar to the animal studies, the subventricular zone has also been identified as being important in human neurogenesis. There are some individuals that believe that in the brain, the cerebral cortex or the neocortex, there is some neurogenesis going on, but as you can see up here, there is solid evidence saying that no, it doesn't occur, and there is also no strong evidence that says that it happens all the time. So again, we're not really sure if this occurs within the human brain. And another area that's found in animals that is thought to happen in humans is the striatum. Again, it's controversial whether or not we have this in humans going on as well. And again, we don't know, and that's sort of bothersome in terms of neurobiology. We should have a better idea of mapping out our own brains first, and there's no consensus as to whether or not these two areas like the neocortex and striatum actually do have neurogenesis or not. Subventricular zone, yes. Dentate gyrus, yes. And a number of these reports actually are in agreement within the human brain as well. So lots of controversies, which is good for you as future researchers, that if you are looking for areas that you need to go out and prove things, this is an area that needs some really solid evidence behind it. So one of the other reasons why we're interested in this entire process, when we look at this process of neurogenesis, which might be disrupted in depression, it might recover after the use of different antidepressant medications, these newly born neurons in that process going from left to right on that previous diagram actually have more excitability than other neurons around it, and they're also much more synaptically plastic. They're much more likely to develop LTP as well as LTD, and these are incredible if you think about what that means. So this is one reason why people think, this one bullet point is the whole rationale for why people think that exercise, which has been implicated in increasing neurogenesis, why exercise is better for your brain in terms of cognition. So it makes sense. I go out and exercise, these newly born generated neurons after exercise might be much more plastic and much more able to integrate and have more synaptic plasticity than the ones that normally are there within the brain. And again, it says that there's a lot that we need to understand about this process. Neurogenesis also declines with age. Again, very controversial. Many of you will have seen the papers last year that say that by the time you're 40 or 50 years of age, you have like no neurogenesis left. And this is why people develop Alzheimer's disease. And other groups came out immediately after and said, nope, that's wrong. We actually detect in older individuals all the way up to the age of 75 that we still see neurogenesis. So again, there's so much controversy in the field that it's really hard to get a good sense as to the importance of neurogenesis. And we need to understand how neurogenesis occurs, why neurogenesis is up for debate. Even though it's a well-established phenomenon, why do scientists disagree about this? And why don't we have reproducibility in every single lab? Yes?
Is there a consensus that neurogenesis
So there is no consensus that neurogenesis continues throughout someone's lifetime. So some scientists say that yes, it does continue, but there's no consensus. Other scientists say that no. After the age of 40, in humans, that there is no continued neurogenesis, or it drops off to such a low level that we can't detect it. So no consensus. Yes?
is there any evidence of difference in neurogenesis process in people who have healthy brain state
So it's a great question. So is there a difference in either the quality or the number of cells that are being generated through neurogenesis in a diseased brain versus like a healthy brain? And there is some evidence to suggest that yes, there's both a reduction as well as a difference in the types of neurons being produced in an unhealthy brain. And this is one of the reasons why the induced pluripotent stem cells, the iPS cells that you can take from your skin, why they can actually model the brain as an organoid and they still carry the disease phenotype. So great questions. So here's one way that we actually go in and we can measure neurogenesis. And these are things that we normally do. At some point, if we think that neurogenesis or new neurons are being produced, we will inject into the animal, so we inject in intraperitoneally, we inject this chemical substance known as BRDU, and BRDU is a thymidine analog. So as DNA is dividing, and DNA divides when the cell divides, and the cell divides when that pluripotent stem cell starts replicating itself, it will take up BRDU. We normally don't have it in our DNA, and we use an antibody to see, are there any cells that have taken up this marker for BRDU? We will not see this in any cell that has not divided. BRDU is only present in dividing cells. The other thing that we can look for is this endogenous marker known as double cortin or DCX. And double cortin or DCX is a marker for neurogenesis. It tells us that a cell has undergone neurogenesis, and it will also tell us that it has migrated. So remember, as I showed you here earlier, in this process, it starts to move and it moves up within this granule cell layer. And so we need to be able to detect this movement or this migration of these newly formed neurons. And it is an important factor to look for. So if we see BRDU and we see double cortin, that is how scientists normally detect for neurogenesis. And you think that these are such an easy marker to look for, why is there so much controversy? And some of it is, some of the numbers and the amounts of the staining are so low that it would be hard for everyone to agree that you have a stained cell or you don't have a stained cell. So this is how they detect it, and these are the methods that they use, and sometimes the numbers are so low to begin with that scientists have a hard time coming up with a consensus for that. You don't have to worry about panel F down here at the bottom, which is marking more of the mature cell markers like the calcium binding protein or CABP. You need to know about BRDU and the function of double cortin and how we measure neurogenesis. Okay, so this also means that if we were somehow able to isolate some of these progenitor cells, or these stem cells, we could actually continue on their production. So they divide, they take up BRDU, they continue this process. Some of them will continue on as stem cells, and this is what some of the research labs here at U of T do. So if you are familiar with the work of either Professor Mooreshead, or if you know about the research done by Dr. van der Kooi, some of the things that they do is to isolate these progenitor or stem cells, and if you take one stem cell and then you provide the appropriate growth factors in a dish, so you would do this in vitro, and some of you have done cell culture before, and if you provide them with factors like EGF, epidermal growth factor, fibroblast growth factor, so remember FGF2 was a molecule I showed you earlier, as well as heparin. If you provide all three of these in the medium outside of the cell, it will continue to divide, and it will actually form what is known as a neurosphere. It will form a three-dimensional structure. It floats off of the bottom of the dish, and you can then divide up this neurosphere, as you are seeing here. So you break up this neurosphere after you have fed it, and after you split up the neurosphere into its individual cells. If this is truly a neurogenesis-like model, you should see each of these cells in turn from this neurosphere capable of forming another neurosphere, which is what you are seeing here. They're capable of replication of themselves. If we wanted to see, are these actually neuronal in origin, one of the things that we do is we add this substance which is known as fetal bovine serum, or FBS. And fetal bovine serum is a different type of trigger, and it causes these neurospheres to differentiate. So if you have these neurospheres and you've removed all of their normal growth medium like EFH, now you add FBS, immediately you will start to see them differentiate. And these neurospheres, because they have the potential to form any of the different types of cells that you would find in the brain, will actually form astrocytes, they will form neurons in green, and they will form oligodendrocytes that you see here in red. So we have ways to identify histochemically that we have neurogenesis going on. We can also look at and characterize the features of these progenitor cells by using this in vitro model and to see if they do indeed form all of the different functions or form all of the different cells that we would expect to see within the brain. So we will have neurons, we'll have astrocytes, we'll have oligodendrocytes, and these are the different markers that we would use to identify astrocytes, a specific marker for neurons, or a specific marker for oligodendrocytes. These are all, again, antibodies that we would use against GFAP for astrocytes, an antibody against beta-3 tubulin for the neurons, or antibodies against O4 or myelin basic protein to identify oligodendrocytes. And we'll see all three of these if we did indeed start off with a neural progenitor cell. Any questions on this before we go on? No? Are we good? Yes. So I'm not sure if I understand your question correctly, Miriam, but I'm going to take a little tangent. So could I get this type of result if I took a stem cell from the subventricular zone? So this is actually studies that are done in the subventricular zone. I should. Could I do the same types of things if I took it from the dentate gyrus? So remember the dentate gyrus, the subgranular zone layer, SGZ, contains these radioglial-like cells that eventually form progenitors. Could I see the same thing going on here? In theory, yes. It's not done as often because it's a smaller structure. It's harder to isolate as many of those progenitors. Yes? Yep. So the question was, if we have ways to expand populations of these progenitor cells, so you can imagine, like, I could start off with one cell and I could get billions. I could form the entire brain if I waited long enough and had enough of these neurospheres. Why don't we have like more replacement therapy using stem cells? And there are lots of issues with it. So the first thing is, if I'm replacing cholinergic neurons, I can't guarantee that these neurons that result here after FBS are going to be cholinergic neurons. I have no way to know that they're going to differentiate into the cell types if I implant them into the area of the brain that I need them to go to. And we have no way to stop their continued division in that local environment. So there are lots of issues that are still associated with stem cells. Okay, awesome. Yeah, no worries. Okay, so eventually we're going to get into the role of exercise. And what I'll tell you is that this is not disputed, by the way. So we're going to talk about controversies related to exercise later. None of this is disputed at all. Like every single lab here at U of T, labs down in the United States, labs in Europe, every single lab agrees that if you have exercise, you are going to increase neurogenesis. The mechanisms, under dispute. Other things, under dispute, but there's no doubt that if you take a normal mouse and you look at the dentate gyrus and you've injected in BRDU, and each of these little black dots that you see here represent a cell that has taken up BRDU and therefore is dividing. So this is a marker for neurogenesis. And then if you examine this 8 weeks later, you'll still see some of these little dark spots, the nuclei of the cells containing the BRDU that we started off with. And so those cells do indeed form, and we do get neurogenesis. If we looked at an animal after exercise, you can see there's a huge number of cells, much more than we would have under resting conditions. Most of these, or there's an increased number of these that also survive 6 weeks later if we exercise. There is no doubt that exercise has an impact on the number of cells that are being formed and that survive with exercise. The problem is we don't understand the mechanism. The mechanism of why exercising produces this effect is completely open. The effect itself is not under debate at all. Okay, so I wanted to make that clear. Do you have any questions on this before we go on? Yes? Sorry, is it fair to say... So your question is, okay, so at the top, we actually see more cells, and at the bottom, we also see that there are more surviving cells. Which effect does exercise have a bigger effect on, if I can reword your question a little bit? So it turns out exercise has an effect on both. And do you remember the two factors that we looked at for proliferation and survival? Without looking back at your notes, anyone? Yes, Isis? BDNF is for which one? Survival or proliferation? Survival. And so VEGF is for increased proliferation. And it turns out that exercise enhances both VEGF and BDNF. Okay? Great question, so thank you. And by the way, knowing that subtle difference, proliferation versus factors that are associated with survival is important. It'll come back to haunt you in your dreams on your final exam. Just telling you in advance. Okay, so eventually we're going to get into the model of like how exercise can affect a number of different organ systems and how all of these organ systems in turn can affect your brain. And we think we understand some of these pathways, but all of these pathways, every last one of these pathways shown here are under dispute. Like there is no consensus. And we're going to go through these in some more details later. And we will talk about all of these different factors. Do you remember this one molecule? Interleukin 10. Do you remember that from an earlier lecture? Is it pro or anti-inflammatory? So this is anti-inflammatory, and so remember the common theme. So does diet induce anti-inflammation? Does omega-3 fatty acids, does it produce like an anti-inflammatory effect? Does exercise produce an anti-inflammatory effect? Do antidepressants produce an anti-inflammatory effect? And so this is sort of the new way that people are looking at this. So I think we're going to do one quick poll. I'm going to take you through things related to BDNF that we're going to need to know. There's a lot that's pretty heavy, and I'm going to take you through this slowly after the break. And I'm going to tell you a little bit about one of the greatest neuroscientists who passed away a few years ago. This is Rita Levi-Montalcini. Again, she was the first woman neuroscientist to win a Nobel Prize. So she was really a groundbreaker. She experienced a lot of bias, and she experienced a lot of sort of things that might have made most people give up on continuing on in neuroscience, particularly early on in her career. And we'll talk a little bit more about her trajectory and how she overcame a lot of these obstacles a little bit later on. And I'll go through these two Nobel Prize laureates, and we'll talk about some of these breakthroughs that have occurred in understanding these neurotrophic factors like BDNF. This molecule is a really cool molecule. We don't understand everything about it, but I want you to understand a little bit more about it if we at all can. So before we get to this slide, I'm going to ask you to go back to your Notonic. And some of you have been asking about this. So just again to go on. So do you know like when you have to submit your assignment by to get feedback? So the latest that we'll take it. What is the latest that we'll take it for you to get feedback? Right. Okay. So I think most of you know we'll take it up to the 20th. It has to be a full draft. And here's why we've been taking our time kind of getting that Dropbox ready for you, and there is actually a reason for it. Some of you have been also asking, when will the marks for the visual summaries be put online? So we're making sure that all of the marks from all the different TAs are more or less agreeing with each other. We don't want some TAs to be marking harder than others. We want some parity between that. So we're just making sure that everyone has a lot of A's or a lot of A pluses, and we don't have too many people marking more harshly than others. We want it to be a fair process. But all of your marks for your visual summary will be up there by Friday. If you don't have your mark up there by 12 noon Friday, let me know and I'll make sure that they do get posted. And the reason why we're hoping that you're going to take your time doing this, oh, sorry, not quite yet, not yet. Okay, go back. Okay, awesome. Let me ask you this. If you're submitting a draft of your paper, it's almost done, you just want some feedback, you're submitting it to Quirkus, it's going through Turnitin, and you think it's a draft, if there are things that are copy-pasted on your draft, and you think it's a draft, and it's being submitted to Quirkus, is that an academic offense or not? It's an academic offense. So just because it's a draft, and you're submitting it and you think that it's not going to be, you're not going to be receiving a mark, that is still considered an academic integrity offense. And I don't want that to happen. So it would be better for you to take your time, make sure that you're not copying and pasting lines of text. And if you're not sure like what that means, or if you're not sure like how to paraphrase something, or if you're taking like a block and you think, and this is the most common thing that I hear, which is, oh, I just couldn't think of another way to rephrase something. That's completely wrong. And even if you put in brackets after what you have just copied over, the citation itself, it is still considered plagiarism. So for example, in today's lecture, if I had mentioned that not Nguyen. So if I had said things like BRDU is a marker for neurogenesis, as well as double cortin, and you use that, and you put like cited by, et cetera, and you used it verbatim, and you said that I could think of no other way to actually put that down, that would still be considered an academic integrity offense. So please make sure that you are aware that when you submit, that Dropbox will be available as of this Friday as well, that when you submit, you should also be checking that you have not copy-pasted, you've paraphrased correctly, you've cited correctly, and the other common issue is sometimes it might be by mistake, and sometimes it's not by mistake, it's because you're rushing and you just want to pad your reference list. So this is called a false citation. You actually read that paper, you want to make it look like it's a better looking reference list, so you just like add a reference and you just stick it in there and you stick it in the context of that particular paper. Turnitin can actually pick up that false citation, and that's also an academic offense. So don't put in things that you haven't read, okay? All right, with all of that doom and gloom, I think we need a little bit of music. Yeah, did you have a question? Yeah, all of the sections should be done. It shouldn't be like, oh, I've done everything except I haven't included like the future directions. So everything should be done, and it should be like 95% complete. We'll get you your comments back for the different sections within a couple of days. All right. So, are you guys ready to go?
It's crazy how fast the shit goes. I cannot be late anymore.
Yo, what's up, guys? My name is Steven. I'm part of your tech group for this course. I'm also part of a club called Giving Sounds of Music. And with this cold weather outside, just want to pass on some more vibes for y'all. That's cool.
My name is Adine. Thank you.
Okay, so now we're performing Everything I Never Before. You're a falling star, you're the getaway car, you're the line in the sand. When I go too far, you're the swimming pool, I'm in love stay, and you're the perfect thing to see. And you play it coy, but it's got you. But when you smile at me, you know exactly what you do. Baby, don't pretend, true, you know it's true, 'cause you can see it when I look at you. And this crazy life, and through these crazy times, it's you. You make me sing your every line, your every word, your everything. You're a carousel, you're a wishing well, when you lie me in. When you're in my bed, you're a mystery, you're from outer space, you're every minute of every day. And I can't believe all that I'm your man, and I get to kiss you, baby, just because I can. Whatever comes our way, we'll see it through. And you know it's what I can do. And in this crazy ride, and through these crazy times, it's you. You make me sing your every line, your every word, your everything. So la 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 la. So la 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 la. And in this crazy life, through these crazy times, it's you too. So make me sing your every line, your every word, your everything. Your every song that I sing along. 'Cause you're my everything. So la 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 la. So la la la
So we're both feeling sounds of music. Thank you so much. Have a good rest of lecture. And thank you, Dr. Drew, for letting us come here.
Thanks for coming in.
Good job, you guys. I love the first song, so I'm so happy to hear it. I'm so sorry about the speaker again. I think
No, it's not. It was like
All right. Okay, folks, if you don't mind, we're going to get going. We still have a lot of ground to cover. There's a lot that I want to get through to talk to you about the roles of BDNF within the brain. There's some key experiments that I want to go through with you related to BDNF in the brain as well. And I want to also go through like what the growth factor receptors are, how they work in a little bit more detail, and the things that you'll need to know for your final exam along the way. Okay. So these are some of the different things that I'm going to try to get through. And the key point here is that BDNF in general, and again, it's a generalization, BDNF is good for the brain. However, the other thing that you should take away from today's lecture is that not all forms of BDNF, in fact, 2 forms of BDNF are not good for your brain. One is known as pro-BDNF. If you have pro-BDNF, it's not good for your brain. Then there's another single nucleotide polymorphism, which is known as MET66 or valine 66. So the 66th amino acid, if you have a methionine or a valine, really alters the function or alters the role of BDNF within your brain. So not all BDNF is created equal. Pro-BDNF is not good, and neither is a single nucleotide polymorphism associated with BDNF as well. Okay, so to get there, we need to understand more generally this concept of what the different neurotrophic factors are. And some of these different neurotrophic factors include this classic experiment that was done by Victor Hamburger. So this is Victor Hamburger down here at the bottom. And this experiment that was done literally in his basement at the turn of the century, like the early 1900s, was actually an experiment that was controversial at the time, and we're going to go through it because it's such a crucial way to look at neurotrophic factors or factors that allow for neurons to become healthy, to divide, etc. So he was using chick embryos. So chick embryos are sort of the preferred model back in his day. So nowadays we use mice, or we use C. elegans, or we'll use Drosophila. But back in the day, chick embryos were the model of choice for many developmental biologists, especially in Europe. And one of the things that makes the chick embryo particularly amenable or useful in these experiments is that you can keep the embryo viable. You can also see it under a microscope. So all you have to do is create a little hole at the top of the egg, remove that eggshell. You can see the developing embryo in the yolk. You can do surgeries on it, and you can cover it back up with like wax paper, and the embryo will continue to develop just as it normally would. So he decided to use this chick embryo, and one of the things that he did was he excised one of the limb buds, so it would normally form like the leg. And again, you have like 2 of these different limbs, one on the left, one on the right. He decided to ablate or remove the limb surgically. This was the surgical tool that was used to remove it. And then 1 week later, he would go in, sacrifice the animal, and then go back in and count the number of neurons that he was able to observe within the spinal cord. So the spinal cord is going to connect to one of these missing limbs. And what he wanted to know was, if I remove the limb that normally connects to this section of the spinal cord, what happens to the neurons? And so what he saw was like pretty dramatic. And he did this experiment. How many times would you do an experiment like this to convince yourself that it worked? 10 times. How about some of the rest of you? 3, 4. So he did this several dozen times just to confirm that this experiment was working, and he saw on every single experiment that whenever he had ablated the limb, on the side that was missing the limb, he would actually have many, many fewer sensory and motor neurons in the ventral horn of the spinal cord. Okay, so he saw this consistently. And so he was thinking, this is interesting. Like, hey, why is this happening? There are a lot of different possibilities for why this might be happening. And he wasn't really sure like why this particular experiment. He knew that it was a consistent result. He knew it could be replicated. In fact, one of the people that he asked to replicate his experiments is the next person that we're going to talk about, Rita Montalcini, who saw his research and wanted to help with his research. So he decided that, hey, if this works the way that I think it's working, I'm going to try like a different experiment. So I'm going to take two animals. I'm going to do that first limb bud ablation that I did before. So I'm going to remove the limb. But instead of just throwing it away, I'm going to actually take that limb and I'm going to attach it surgically to this developing embryo near where its normal limb bud is. So it's like having like two legs in the same side of your body. Then 1 week later, he goes back in, and he saw that the transplantation of a supernumerary limb or an extra limb actually resulted in an increased number of these sensory and motor neurons. So take away a limb, you get reduced numbers. Add a limb, and you get an increased number. Does that sound reasonable to you? Okay, so this is one of the things that he's kind of thinking about. So from both of these experiments, but particularly the latter experiment, one of the things that he was thinking, this is his original hypothesis, and you can go back and read his 1929 manuscript, is that he thought that within the spinal cord, there were these undifferentiated cells which could develop into other cell types or cause proliferation to occur. So the targets, the limbs, were actually recruiting some of these undifferentiated cells, the ones that weren't like going to be sensory or motor neurons, and they were changed into more sensory motor neurons, sensory or motor neurons. And it turns out that this hypothesis was wrong. So it's not about recruiting undifferentiated cells. His hypothesis, even though he had published this as a hypothesis, actually turned out to be wrong. And as he published it, and this is why we do this in science, although now in science it isn't about getting your ideas out there and getting confirmation. It's about getting validation for your experiments. And science used to be very different back in his day. So he got his hypothesis out there. People started to do and replicate his experiments, but people started to realize this is not undifferentiated neurons sitting within the spinal cord being recruited to become more sensory neurons, or not being able to be recruited, and that's why they disappeared. There was some kind of survival factor that they were hypothesizing. And in 1942, other groups, based on his original publication, started to come out and suggested it's not that you have more differentiation or less differentiation if you remove a bud. It's that there's some kind of unknown target-derived factor, and that's how they described it. It's an unknown target factor within the limb that is keeping these cells alive. So we need it to keep cells alive. In retrospect, you learned all of this, the summary of like 100 years of neuroscience, in the previous lecture that we had, where we look at all three of those different pathways that get turned on after BDNF. That's the sum total of like everything that we've learned since Victor Hamburger back in the 1920s. And so with Rita Levi-Montalcini, who had read his paper, who decided to work with him, both Victor Hamburger as well as Rita Levi-Montalcini found that there was indeed more death. So what they were able to see is that without this limb, there were more cells that were dying, and that with an extra limb, there was more factor around and there was less cell death. This was considered, this paper in 1949, which definitively showed that it was cell death. A factor from the limb is producing a factor that stops cell death from occurring. This was considered to be a groundbreaking paper. Now keep in mind, this groundbreaking paper that has revolutionized our understanding of how the brain works, how developmental biology works, was published in the Journal of Experimental Zoology. So would you want to publish in 2019 in the Journal of Experimental Zoology? I would argue probably not, because science was very, very different back then. It wasn't about high-profile journals like Nature or Science, although they still existed back then. It's just about getting your research results out. And this is the beginning of what is now known as the neurotrophic hypothesis. And if we take this one step further, Do you remember that third pathway that we talked about? So BDNF or some other growth factor binds, the receptor dimerizes, we turn on the MAP kinase pathway, we also turn on the phospholipase C pathway. Do you remember the other pathway that gets turned on? Sorry? So eventually, through that pathway, we will activate PI3 kinase. And the activation of PI3 kinase and AKT kinase, what does that stop? So I had mentioned at the time that apoptosis, these neurons, your neurons, every last one of your neurons in your brain is basically just waiting to die. Like without BDNF there, that pathway, that AKT pathway that stops caspases from being activated, that's going to be turned on. And so this neurotrophic hypothesis is actually a really key pathway for us to start to examine. There is a lot of proof in 2019 that this is actually correct, and I'm going to take you through some of the data. None of these dates are ones that you need to know or memorize. There's actually nothing on this slide that you need to memorize. I do have a question up there, but it's more to just see if you know what this explant culture is. So this is the experiment that they started to do when they identified this unknown factor. So they reworked their model. So the original model was this chick embryo in an egg, and they decided that, okay, this is good, it's a great model, but we're going to switch models. It's like moving from C. elegans to Drosophila, or moving from Drosophila into mice. We need to use a different model. And so one of the models that they decided to do, they needed to still work within the nervous system, because that was important to them. They wanted to use what is known as this explant assay that you see here at the top. So they took a chunk of the nervous system, they put it into culture, and they wanted to know if we added different growth factors to the medium, the cell culture medium that is bathing this chunk of tissue in black. So this is a chunk of tissue. Will it have neurites that grow out of this chunk of tissue? So one of the things that you need to have is like neurite outgrowth for this growth factor to work. And this became known as the neurite outgrowth assay. So do you know what tissue they use for the neurite outgrowth assay? Subventricular zone. This is like long before they identified adult neurogenesis. Any other guesses? What would be like easy to surgically remove in the 1950s? Part of the nervous system. Yes? Squid giant axon. Good guess, but not squid giant axon. So they're still using chick, by the way. So this is still chick, but they're not working on the whole body. They're working on a specific. Spinal cord would be a good guess too. So this is the ciliary ganglion that controls like the eye. And so this is a ciliary ganglia that they've explanted. And again, the details are completely irrelevant. You don't need to know about any of this. And so using this chick ciliary ganglia, and by adding like different factors, so they tried it like systematically, like literally they would have like all of these cultures set up, and if you go back and you look at their original papers, they'd have like a row of all of these different cultures, and for each of them they would have like different chemicals that they would add to it, and they would try different combinations of these chemicals, and eventually, and looking under a microscope, they knew that they would have the right molecule when they saw the ciliary ganglia have all of these neurites growing out. So when they have a growth factor, this is what they would expect to see. If the growth factor isn't very good, then this is what they would expect to see. And eventually, through trial and error, and by trying like different combinations of ground up tissue, they were able to find by 1960 that there was a factor that they called nerve growth factor, or NGF, that they were able to purify it, not to the standards that we would use today in 2019, but they were able to show that it was about 98% pure NGF. There were still other cellular contaminants, because remember in the 1960s, they don't have like recombinant DNA technology. They're literally taking parts of brain, mashing it up, running it on HPLC, trying to get different fractions, isolate those fractions, and then apply them to this chick explant. Eventually, and this is sort of the sad part to this story, so Rita Levi-Montalcini, who single-handedly by herself convinced Victor Hamburger that his theory was wrong, and allowed for the development of the neurotrophic hypothesis, was working on this new assay that she developed, and she's developing and working on this, and by herself she is kind of getting closer and closer to purity of this factor, but she has to get help from a biochemist by the name of Stanley Cohen. And so even though she had done the vast majority of the work, the theoretical hypothesis, modeling, etc., she shared the Nobel Prize with Stanley Cohen because of his biochemical expertise in being able to purify NGF to complete purity. Like there are no other contaminants around. So again, kind of an interesting sort of segue into how Nobel Prizes were awarded back in the earlier times. But she is still the first female that was even considered for the Nobel Prize for her work. And she wasn't awarded the Nobel Prize immediately, like it took decades for her work to be recognized. So this is an important slide, as is the next slide. So I want you to circle this one. If you know that you're not so great at memorizing things, these are a group of slides that you probably want to circle. And so from that time, NGF was the first identified neurotrophic molecule that fits the neurotrophic hypothesis. And this, since that time, we have determined that within the brain, there are probably other neurotrophic factors as well. And we call these neurotrophins. They're a family of things that look like NGF. So all of the neurotrophins, including NGF, are synthesized as precursor molecules. And these are about 250 amino acids in length, and then they are further cleaved, processed by different enzymes to about half their size, to about 100 to 120 amino acids in length. So they look a lot like hormones, in other words. So when you learn about proinsulin, and proinsulin gets converted to insulin, the growth factors are very similar to that. So NGF is the prototypical nerve growth factor, and this is what a prototypical growth factor has to be able to do, and this is why it's prototypical. BDNF has to do the same thing, NT3 and NT4/5 all have to do the same thing. It has to maintain cell survival. Without this, the cell dies. So without NGF, that ciliary ganglia would die. It would have to cause more dendritic or neurite outgrowth. And BDNF has to do the same thing. It has to allow for more dendrites to form, and I showed you that in terms of the context of depression. Strangely, we don't actually need this in the brain. We don't have a lot of NGF within the CNS. We need it in the peripheral nervous system, even though it was isolated in this particular fashion. The one that we are talking about, BDNF, brain-derived neurotrophic factor, is in the same family, and it has about 50% homology, so it's about 50% identical to NGF. Originally, it was purified from pig brains by Hans Tonen in Europe, and again, NT3 and NT4/5 were not isolated by pure biochemistry, so they took brains, they basically homogenized the entire brain, ran it through an HPLC, got the different fractions just like Rita Levi-Montalcini did, and they used the same techniques in identifying BDNF back in the 1990s. But later, when PCR cloning became available, by position cloning, NT3 and NT4/5 were identified as other putative neurotrophic factors within the brain. So why is all of this important? So we have this prototypical neurotrophic factor, NGF. We have these others that are known as BDNF and NT4/5, as well as NT3. And then afterwards, much later, we were able to identify their receptors. What do they actually act upon? And so NGF will act only, and NGF only acts on the TrkA receptor in this particular diagram. So NGF has like very strict control on what it can bind to, and it binds to the TrkA receptor. Going back, if you were to describe this, what kind of receptor are you looking at here? Is it a monomer or a dimer? So we have one monomer, the other monomer, so this is dimerizing. The tyrosine residues would be found intracellularly. BDNF will bind to the TrkB receptor, but so will NT4 and NT5. So again, make sure that you realize that TrkB can actually bind BDNF, as the name would suggest, but it can also bind to NT4 as well as NT5. Two totally different neurotrophins can bind to the same receptor, and NT3 can also bind to the TrkB receptor as well. Strangely, NT3 can also bind solely to the TrkC. BDNF and NT45 cannot bind to the TrkC receptor. There's one other little caveat. There is another type of receptor for these growth factors, which is known as the P75 low affinity neurotrophic factor receptor, or LNTFR. And it's a weird receptor. It does not look at all like the receptors that we see here. So all of them are monomers. We have them dimerizing. We have tyrosine residues intracellularly that autophosphorylate after binding of the neurotrophins. But this low affinity nerve growth factor receptor, the P75 LNTR, is a little bit different. And if we were to take a look, and I think I have this on the previous slide, it does not look at all like these types of receptors. It looks more like the tumor necrosis factor receptor. So the TNF receptor is much more homologous than TrkB is to this P75. Again, really, really interesting. And here again is why we're interested in this. This is one of the receptors that you see here. So this is one unit of the P75 receptor. And if we take a look at some of the different domains that we see intracellularly, there are no tyrosine receptor molecules that we see. And instead of promoting cell survival, if a factor binds to this low affinity growth factor, it actually promotes apoptosis. And it makes sense. They contain a number of these death domains that you see here, these type 2 death domains that recruit other apoptotic signaling pathways. And again, you might think, well, why is this important? Because remember, we have two different flavors of these neurotrophins. They all, every last one of them, begin as larger versions of themselves. So we have NGF that begins like at 250 amino acids. It gets cut in half to about 120. And we also have BDNF, and we have like NT3, NT45. All of them begin as bigger molecules. These bigger molecules, which we call pro-BDNF or pro-NGF, etc., and in particular pro-BDNF, this can bind to the low affinity growth factor receptor, and this pro-BDNF, if the receptor is around, and if we have pro-BDNF around, actually induces cell death. So when we talk about BDNF, not all of it is good. We need to process BDNF appropriately, or it can actually cause cell death through apoptosis. The large version of it is not a good form of the molecule. So again, BDNF is a good molecule, causes cell survival when it's interacting with TrkB. As per the previous diagram, we know that things like NT45 can also bind to BDNF, so can NT3, the TrkB receptor. And pro-BDNF will actually cause apoptosis through this low affinity receptor that you see here. So it's a confusing pathway, but it also kind of makes sense that we need a way to regulate this cell survival. So this is a really busy slide, and I don't need you to memorize all of this busy slide. It's just to integrate some of this for you. There's nothing on this slide that you need to memorize. I guarantee you nothing. Just here to kind of give you a roadmap for why we're interested in this. And one of the things that we know, BDNF comes from neurons. And neurons can actually release BDNF. And BDNF can bind to things like TrkB. And it might help this neuron that has bound the BDNF to undergo those signaling molecule pathways like AKT and cell survival, the ERK kinase pathway that causes like increased cell growth as well as activity, and it can also cause the transcription of even more BDNF in this cell to cause cell survival. So it's a good thing for all of this to occur. So this is, again, these next two slides are really busy slides, but you need to know them, and you need to know about the pathways. I can also tell you your quiz questions, as well as the multiple choice on your final exam will come from this slide and the slide that accompanies this on the next diagram. So if you have questions along the way, please feel free to ask. So I told you earlier in the previous slide, all neurotrophins begin as larger molecules. And what you're looking at here is the larger version of the molecule, and this is pro-BDNF. Pro-BDNF is found in these vesicles that come off of the Golgi, and if it is converted appropriately within this vesicle by a molecule like furin, intracellularly it can just be released from the cell as the mature version of BDNF. So furin will cut off this little excess, and it will convert it from 250 amino acids to about 120 amino acids to the mature good form of BDNF. It can also undergo regulated secretory release. So this is activity dependent. Every time a neuron is releasing neurotransmitter, it releases things from vesicles. It can also release BDNF from vesicles. So this is that particular pathway. But we don't want to be releasing any of this pro-BDNF. So every time it goes into this regulated secretory pathway, there's an enzyme called proconvertase. And on the next diagram, it's called PC1, and it's proconvertase 1. So proconvertase will take it from this vesicle, and it will remove that larger domain to make the active form of BDNF that can get released from a neuron as well. So we have different versions that can get released from a neuron. So the furin pathway releases mature BDNF. The regulated secretory vesicle pathway through proconvertase 1 or PC1 will release BDNF. And if these pathways aren't around, this neuron could also release pro-BDNF. So all of these pathways are in place. So pro-BDNF, is it the end of the game? Well, no, not necessarily, because associated with neurons on their membranes, we also have another enzyme which is known as plasmin. And plasmin is also a protease that can convert pro-BDNF that gets released from this neuron into this mature version here, and it can work the way that we would normally expect. Activate tract B, activate all of the good pathways that we talked about. Pro-BDNF could, in theory, work on its low affinity receptor. It could be internalized into the cell. It could stimulate the death pathway, but again, it's not all over here as well. There are mechanisms that we don't know fully yet that can also convert some of that pro pathway, if it gets internalized, into the mature version as well. Again, it's not really clear what pathway 5 does yet, and people are trying to figure that out, but it is possible that all of these are occurring at different stages. So on this particular pathway, all I really wanted to get across is that this pro-BDNF is not good. Normally it binds to the P75 receptor. We need to convert it either through the furin pathway or the proconvertase pathway, so either pathway 3 or pathway 4. We get the release of mature BDNF. This is the version that we want, and this is what most neurons like. And even if the pro-BDNF gets out, this plasmin, which is sitting on the neuronal membrane, can also convert this as well. So we need to have this regulated release of this BDNF. So I just mentioned a whole bunch of things to you over the last few minutes. Do you have any questions about this before we go on? Yes? Is it always not the worst case scenario? It's not the worst case scenario because we have so many pathways to convert it to BDNF, which is what we want. But if we have it, it's not a good thing. Anything else I can answer for you now? No. Yes? It's the pro version, the larger versions, not the active forms, not the shorter forms. The pro versions, the longer length versions. Yes, all pro neurotrophins. Any other questions? No? So BDNF is, I'm going to try to convince you over the next few slides. None of these are important slides that you need to memorize for size effects or anything else. If we have BDNF around, and the increased amount of BDNF will actually enhance neuronal function, so we get better EPSPs, we get better responsiveness, we get more spines being formed, we will have larger spines with more BDNF, we have increased synaptic plasticity with more BDNF around. BDNF is good for your neurons. Not the pro version, but the mature version is incredibly good for neurons, it's incredibly good for spines, it's incredibly good for neuronal survival. All of it is really necessary. So the more activity that we have, the more of that regulated pathway that uses proconvertase, regulated secretory release, more BDNF gets released at this synapse, which is what you're seeing here, that regulated release pathway. And that means that we get more BDNF being released onto this particular spine. That means that this neuron is going to receive a lot more BDNF. So this, in turn, will make this postsynaptic spine that you see here much more likely to undergo synaptic plasticity. There will be more AMPA receptor stability, and AMPA is a glutamate receptor. And you might also get more production of nNOS, or nitric oxide synthetase, which allows for a retrograde signal to go back to signal for even more BDNF. So it's a positive feedback loop as well. And it all says BDNF is good for your brain. So are you with me so far? Have I convinced you that BDNF is good for your brain? No, like a lot of stunned silence. Is it good, not good? Good? Okay, I get a very weak, good, not enough BDNF out there. I'm hoping that I've convinced you that BDNF is a good thing. And I've told you that there are two versions of BDNF that are actually not so good for your brain. One of them is pro-BDNF. Pro-BDNF is not good for you. The other version is the scenario that I'm going to mention to you next. So taking in mind that I've just tried to show you that BDNF is good for spines, plasticity, good for neuronal survival, maturation, all of those things, in the beginning of the genomic era, so right at the start of the genomic era, people were particularly interested in the genes that were associated with how your brains function. So your brain is always a place where genes are examined to look at function. So it'll continue happening until we figure everything out. And we're a long way from being able to do that. And so one of the very first genes that was looked at was BDNF. Like everyone knew by 1998, 1999, around the time of the human genome sequence being put out, that BDNF was a molecule that our brains needed for proper development, proper functioning, survival, maturation, all the things that I just mentioned. And people started to identify that consistently within families that there was one polymorphism that always stuck out. And there was a polymorphism in the BDNF gene that resulted in either having methionine at position 66 or valine at position 66. And depending on which version of this you had, you would actually have decreased secretion of BDNF. And that meant that you might have less BDNF being released, less BDNF meaning less synaptic plasticity, less synaptic plasticity meaning worse memory, worse learning. And it was also thought to underlie having less BDNF being released by having a polymorphism also resulted in having increased anxiety as well as depression. So we made that full circle back to BDNF and depression. Additionally, BDNF was also known by the 1990s that if you block BDNF signaling by blocking with an antibody that will use the same binding position on track B. So the track B is the receptor. By blocking BDNF signaling, the number of synapses will decrease. So it's like BDNF is not around because it's not able to bind to its receptor. And that exercise-mediated increase in memory, which is, again, agreed upon universally, if you have anti-track B receptors after exercise, all of those exercise-mediated effects all disappear, suggesting there's a link between exercise and BDNF. And other proteins like NCAM180, NCadherin, and AMPA receptor complexes, all of these things that are part of the synapse that normally expand after exercise and after BDNF is around, all of these increases in NCAM, NCadherin, and AMPA receptors, all of those decline if we block BDNF function. So this is particularly the case where we see all of this occurring within the hippocampus, which all of you know is important for learning and memory. And so by the time the human genome sequence was put out, a number of groups had actually said that the more activity your neurons in your hippocampus have, the more BDNF gets released, and maybe this BDNF release is important in terms of your learning as well as memory. So again, this single nucleotide polymorphism was well established. So one of the first hypotheses with this, because again, late 1990s, early 2000s, schizophrenia, one of the worst sort of mental health disorders or mental illnesses, affecting 1 in 100 individuals. So it's a very, very common condition. And one of the things that happens in schizophrenia is the hippocampus, learning, memory, episodic memory, all of these are affected in individuals with schizophrenia. And since we now have a target protein that has a genetic identifier, where there are polymorphisms where you either have methionine 66 or valine 66, and we have this condition where memory is impaired in schizophrenia, one of the first studies in 2001 to 2003 wanted to examine if schizophrenics have this single nucleotide polymorphism that affected BDNF. It would make sense. Schizophrenia, it's a developmental condition. We see it kind of in the teenage years. It probably starts earlier. We're not really sure, but maybe this link with proper neuronal development, the learning disabilities that we see, maybe they're kind of linked together. At the time, one of the largest studies, we'd kind of laugh at this now because we routinely see studies that are published that have like hundreds of thousands of individuals, but at the time, 641 individuals that were recruited to this study by Egan et al. In 2003 was one of the biggest studies that had been published up until that particular time. Nowadays, if we were to use GWAS studies, we'd have both the mother, father, as well as the affected individuals. We would use trio genetic studies, but at the time, all they did was examine individuals who were affected and their normal siblings as well as normal controls. So here's this theory. I can go out in the 2000s, I can do genotyping, and I can genotype specifically to whether or not you have methionine 66 in BDNF or valine 66 in BDNF. So without going any further, do you have any questions or things that are not clear for you so far? We're good, okay. So here's this really interesting study, and this is where, again, controls in science are incredibly important. Their hypothesis is that if you have schizophrenia, the diagnosable, easily diagnosable mental health condition, that if you have one version and you have schizophrenia, or you have schizophrenia, you should have one version, and normal individuals should have something completely different, okay? So you can get genotyped, people in your family can get genotyped, and what they decided to do was that individuals who, and here, patients are down here at the bottom, they genotype them. Did you have valine, or did you have valine and methionine, or did you have 2 copies of methionine 66 on BDNF? And patients, when they were looked at for their ability to do recall, so learning and memory, regardless of their genotype, they sucked. They just did not learn. Like, you were given like a string of like 10 different objects, then you were asked to memorize them, then you were asked to recall as many of them from a group as possible, and they just performed terribly on this episodic memory task. Not surprising. They have schizophrenia, they're diagnosed with schizophrenia, and memory is affected in schizophrenia. Has nothing to do with their genotype. And so their whole hypothesis has gone out the window, right? So how were they able to publish if all of their data shows that regardless of genotype, whether you have methionine or valine, that you just do terribly if you're schizophrenic on this memory task? There's no difference. Like, whatever genotype you have is the genotype that you have. The problem is not here. The problem is in normal individuals that carry one variant of this, and this is where the control became important. It wasn't the test group, the schizophrenics, that were important. It was the unaffected siblings, so the siblings, as well as normal individuals that were unrelated. They were also tested. They were genotyped, and if they had valine or if they had methionine, they also underwent memory tests. And these controls were actually the things that gave the positive result. And this is why controls in science are important. So if you have 2 versions of valine, regardless of whether or not you have someone in your family that has schizophrenia, you have a better memory because your BDNF is a better version of BDNF than someone who has methionine at position 66 of BDNF. And if you look all the way over at the right, their ability to recall is much more impaired, and it's closer, even though they don't have schizophrenia, to the affected individuals. So first, you all see that there is a reduction in their ability to learn as well as to remember. So methionine 66 is not a form of BDNF that you want to have. Clear? Right. Okay. So again, this was really, really cool because they also decided that, hey, you know what? There's also this brand new technique that has been developed that no one's really published a lot on, which is called MRS spectroscopy. So it's like MRI, but they're looking at specific molecules. And they wanted to know, is having this alteration in BDNF, sure, it messes up your memory, and it doesn't matter if you have schizophrenia or not. Like, if you have methionine 66, you're just not going to be able to memorize as well as someone that has valine 66. Does it also affect the health of these neurons? And you have to wonder, you're not going to donate your brain because most of the time you need it. So what do you do to examine the health of your brain without having to remove it? And so they decided to use this MRS technique. And so they were using a marker known as N-acetyl aspartate. And what they found, this is normally a marker for healthy neurons, and this marker for healthy neurons in MRS was found to be lower in individuals that had a copy of methionine. So if you had valine, you had nice healthy brains, healthy synapses, and if you had methionine, you had unhealthy looking synapses using this MRS technique. So again, this technique allows you to visualize, using an MRI machine, N-acetyl aspartate, a marker for neuronal health, and it looks at the protons in this molecule. So this is the different protons, and for the protons that you find in NAA or N-acetyl aspartate, you get this really nice peak. And if you have two versions of the valine, this peak is like really, really high, and if you have methionine, this peak is like much, much lower down. And how do you know? Like, individuals are varied from one another. How do you have normalized controls? Well, these two molecules, choline and creatinine, are generally found in all neurons all the time. It's independent of neuronal health. As long as you have neurons, you will always have the same level of choline and creatinine. These stay the same regardless of your neuronal health. This is the one that indicates whether it goes up or down in comparison to these. And so this is what they decided to do, that when you look at individuals using MRS spectroscopy, that in comparison to either creatinine or choline, that individuals that had methionine had much, much lower NAA levels when they normalized it, saying that they had unhealthy synapses and that BDNF is important, but the form of BDNF is more important than just having BDNF around. So if it were up to you, would you rather have more methionine or valine versions at position 66 of BDNF? Valine. So let me ask you a different question. So again, like one of the things that I bet you about a third of you are thinking about this right now. I wonder which version I have. Are any of you kind of wondering? So can I tell you the answer? Yes, okay, awesome. The mere fact that you're in a post-secondary educational system, U of T, and let's not worry about U of T being U of T, but U of T, you're here. The fact that you're here at U of T means that you all have valine 66. Pretty remarkable, right? You might struggle, but in fact, the fact that you're here and that you got into university with like 99% certainty, you have valine 66. Yes, you can like make that into a shirt. I have valine 66 BDNF. Sorry? So it turns out if you have 2 copies, it's better for you, but 1 is enough. Yeah. Any other questions? I think for today we'll just sort of stop here. We'll come back and talk about more on BDNF. Have a great week. If you don't see your mark for the visual abstract, please let me know on Friday after 12 noon.